Here in Tonga, some of these islands are uninhabited, some of the nearby islands are inhabited, special marine protected areas. So there's a huge interest in sort of the fisheries species in and around these reefs. What we're focusing on here on the third leg of our expedition is population genetic collections of certain organisms to tell us a little bit about how they're connected to different islands around the South Pacific and how different impacts might play out in the future.
Today we're looking for two types of clam species. We're looking for Tridacna maxima and Tridacna spumosa.
The leg before us, they were doing some photogrammetry of the reefs. That is to create a 3D model of what the reef looks like in the reef structure. We can actually go into that image and identify individual clams that we're after.
Ready?
Ready?
Yeah. 3, 2, go. The particular clams that we're focusing on here, they harbour the same photosynthetic organisms that corals do. So they're able to generate energy through sunlight. You might have heard of coral bleaching. Clams can bleach as well. So when the conditions get too hot or unfavourable, the clam tissue will go from that beautiful changing colours to just plain white. So they are an indicator for climate change. We take small tissue samples from them to do the genetic analyses.
Once we've got the sample, we take a photograph of the clam and the surrounding reef so that we can locate it in the photogrammetry plots.
We'll be able to then tell the distances between each clam within that model and also how genetically different they are as well. We can extrapolate the findings out to similar species in similar parts of the world and tell a little bit about what might be happening at a global scale from results found at this local scale here in Tonga.
So these ones you can make.
Similar to the clams, urchins is another species which is of fisheries interest. The collector urchin tends to live in sort of back reef habitats up close to islands on really shallow reefs. So what we're doing now, rather than diving, we're just snorkeling, looking around in the shallow reef flats where there might be the seaweed that the urchin likes to eat. What we're trying to understand here is if the populations that are here are a bottlenecked population at the end of the dispersal kernel, or whether they're one of the populations that's seeding all of the other parts of the Pacific. The tissue that we're after is the gonad, because that holds the freshest, youngest DNA, and that then goes into our 100% ethanol here and is preserved for the genetic analysis when we get home. The kind of science that we do is the quantitative end of science. So what that requires is lots of samples to say things about the natural environment that wouldn't occur by chance.
Good haul.
And so what we're doing now is just filling out those numbers. Once we leave this expedition, 2 years of planning, an enormous amount of effort on the behalf of many, many people comes down to what we have in these tiny little vials here. The package of information we hope is something that is really meaningful for Tonga and meaningful for lots of the different ministries which we've engaged with throughout this process.